So here we'll be looking at genetic engineering, at least the intro to it. So when we look at our human genome, we have 3.2 billion bases. And those bases are the base pairs, like A, T, C, G, the nitrogen bases. And so when we look at all of our DNA, there's actually only a small section of it that is actually our genes. There's huge amounts of our DNA that are non-coding sections of our DNA. And so, for example, a gene might code for insulin. So when we talk about genes, what we're talking about is the area of our DNA that has the directions on how to build proteins in the process of protein synthesis with transcription and translation. And so scientists, though, how can they isolate just that gene and make copies of it? So if they're trying to work on insulin for some reason, how do they get just that piece when we have 3.2 billion bases, right? So scientists, when they want to work with a gene of interest, they need to make multiple copies of that gene. We call this gene cloning, and they're going to use bacteria to do this. So here's a bacteria. Now, inside bacteria, they have little circular pieces of DNA called plasmids. So again, this little circular piece is what we're going to work with genetic engineering. So to clone pieces of DNA in the lab, so if I was a researcher and I wanted to work with insulin, I could take the gene that codes for insulin and insert it into a plasmid. And then I'd have that bacteria make copies for me of that gene. So as a refresher, when we look at bacteria or prokaryotes, this yellow thing right here represents its main chromosome, one circular chromosome, and these little tiny red circles are the extra pieces of DNA called plasmids. So here we go. So plasmids, they do have a small number of genes on them, outside of that bacteria's main chromosome. Okay, so now we're going to look at making recombinant DNA. So with the name recombinant, it kind of sounds like we're going to be recombining DNA together. So what happens is we're going to take the bacteria's plasmid and then DNA from another source, whether it's from a human, a fish, a plant, fungus, doesn't matter because the genetic code is the code for all life here on Earth, so it doesn't matter which source we get it from. And so what's going to happen? Scientists need to somehow join these two pieces of DNA together. So here's what we do. We take our plasmid and the human DNA. And when we look at this, we see that they're all made of A, T, C, and G. And we're going to use these things called restriction enzymes. Now, restriction enzymes are pretty cool. They cut DNA at a very specific sequence. So this restriction enzyme, for example, anytime it's working with DNA that goes with GAATTC, it's going to cut it. So in the DNA, this area would be called the restriction site. So up here, I can look for GAATTC, and I see it right here, GAATTC. I also see it over here, GGAATTC. Ooh, now in my plasmid, GAATTC, I also see it there. So we have these restriction sites located within the DNA, and if you think about it, with that 3.2 billion bases, this short sequence of 6 letters, GAATTC, is going to happen so many times. And so when we look at the restriction enzyme, what it does is it cuts the DNA, it breaks the phosphodiester bond in the backbone, as well as breaking those hydrogen bonds. So you can see here, this restriction enzyme cuts like this. So now, when we cut it, it kind of goes just like this and cuts through that. So over here though, now you can kind of see I kind of just made those extra pieces just kind of disappear for right now. So what I have here is I have an overhang kind of, right? So what I can do is I can take and find that same restriction site in that plasmid, and also use the same restriction enzyme that's going to cut it. And now I have the plasmid opened. Now, if we just look at the pictures, we can see how this A will pair right here. And then we have TT will pair here. So this end has now been paired to a plasmid, and on this end I have TT, A. So then this end here would be paired to the plasmid. So then when we incorporate this together, what I can do is I can look at these sticky ends. Think of them kind of like Velcro. They match. They're going to base pair together. So here I have some sticky ends that are going to base pair together based on base pairing rules. And I can incorporate that section of DNA from another source into that plasmid. Now this plasmid is recombinant DNA. It's DNA that's from a bacterium and human put together, all because of those sticky ends that follow base pairing rules. So now I have a recombinant plasmid. And so this light blue section here is what I have in this area. So I have DNA from one source and DNA from another source put together. Now the coolest thing about this, sorry I hit pause in accident, is that when we look at the genetic code, the bacteria's enzymes can read human DNA as if it was their own. And so when we look at recombinant DNA and how we make it, first we're going to cut the DNA that has our gene of interest with the restriction enzyme. Then we're going to cut the plasmid with the same exact restriction enzyme. Now the key for this is so that the sticky ends match. We want those sticky ends to be able to base pair together. And then the sticky ends will base pair, and then we'll use the enzyme DNA ligase, and it'll seal those fragments. So now here we have a piece of recombinant DNA, and we can call this a vector because it's kind of like carrying our gene of interest. Awesome. So when we look at restriction enzymes, there's a couple different kinds of restriction enzymes, and the significant thing about them is they each cut their own restriction site. So EcoR1 will always cut GAATTC. When we look at HIND3, it'll always cut the same restriction site. Awesome. And then HIND3 will always cut AAGCTT. Anytime it comes across that in a DNA sequence, it's going to cut that there, and you'll produce sticky ends. And then BAM3 will have its own sequence that it cuts. So each of these restriction enzymes are very specific to the sequence that they cut. So if I want to insert human DNA into a plant, or into a bacteria, or into a pig, I would have to use the exact same restriction enzyme if I want my sticky ends to match. And so each restriction enzyme has their own specific sequence that they cut. So it's kind of like those cool craft scissors that have, if I wanted to put two pieces of construction paper together that were cut with different craft scissors, they wouldn't fit up, right? So I'd need to cut them with the same exact one. So kind of like, if I make the analogy, they would have different restriction sites and therefore different sticky ends. So if I tried to put them together, it wouldn't match. Now, where did these restriction enzymes come from in nature? Like, why do they cut DNA? Well, the cool thing is, in bacteria, when bacteria get infected by a bacteriophage virus, they actually have restriction enzymes that are kind of going to be a little bit like their immune system. They're going to try and chop up that virus DNA so it doesn't infect them. So that's pretty cool. Now, when we look at sticky ends are going to have, like, both ends are going to have sticky ends. I don't know how you say that. So then you can attach them together. So when you have DNA from two different sources, when you use the exact same sticky ends or restriction enzymes, then you have matching complementary sticky ends, and now those strands will base pair together. Okay. So now why do this, right? So when we look at recombinant DNA, we can then insert that plasmid into a bacteria, And then, oh, check this out. So here we've taken that recombinant DNA and we're going to insert it into a bacteria where RNA polymerase is going to transcribe. So there's your messenger RNA. And then you'll have a ribosome come and translate it. So now we have the protein. So with this, you have bacteria transcribing and translating, basically making proteins from a different source. So if this was insulin, was what I was interested in studying, what I have here is I have this bacteria. I'm going to take that human insulin gene, insert it into a bacterium, have their RNA polymerase transcribe it, and then their ribosomes translate it, and now I have insulin made. Now it's important to remember, though, that bacteria don't do RNA processing. Bacteria don't cut out introns or exons. So if the human DNA or human gene you're interested in contains introns or exons, you'd kind of have to edit that out first. All right. And that brings us to our lab in class.